Now moving to gram-positive and gram-negative bacteria. See, this grouping is based on the reaction of bacteria to gram stain. What do we do in it is, bacteria are first stained with weakly alkaline solution. This alkaline solution is of crystal violet, or we say Grenadian violet. And all of them will pick color. So all turn blue violet in color. They are then treated with 0.5% iodine solution. Clear? After doing that, followed by washing with water. Now what happens? After that, we definitely treat them with absolute alcohol or acetone. Here we receive two categories: bacteria which will retain color and which will not retain color. Accordingly, we divide them. Bacteria which will retain blue purple color and which do not retain blue purple color. These two belong to two categories. These one are gram positive bacteria and these ones are gram negative bacteria. Alright, gram positive and gram negative. This example is E. coli, quite famous. E. coli, Escherichia coli, and this one is Bacillus subtilis. These are the two examples. Now, washing the stain in gram negative bacteria is due to high lipid content which gets dissolved the organic solvents. Basically क्या होता है, जब हम washing करते हैं ना gram negative की, due to high lipid content of the cell wall, it is normally dissolved in organic solvents like acetone, so no color is traced back. This is the difference. Alright, now moving to the components of the bacterial cell and the further about the bacterial cell or the prokaryotic cell. We were writing their general features. Then the next one is, these cells, the prokaryotic cells, are generally smaller than eukaryotic ones. Next is, these multiply faster. Next one, all prokaryotic cells except mycoplasma. This is an exception. They carry a cell wall outer to the cell membrane. So have a cell wall outer to the cell membrane. Then another important feature, that in addition to chromosomal DNA, these contain small circular DNA which is known as plasmid external to the chromosomal DNA in the cytoplasm. And a plasmid has genes for certain unique characters. What kind of unique characters? Like resistance to antibiotics. This we are going to study in detail in class 12, biotechnology chapter. This chapter is mainly based on how do we make plasmids resistant to various antibiotics and how do we make recombinant DNA using E. coli. So this is all related here. Okay. Then we move to the main one. It's envelope and modifications. Cell envelope and modifications. Now when we talk about cell envelope and modifications, we say cell envelope, this envelope, it is a tightly bound three-layered structure. So what are the three layers? The first one is the outermost glycocalyx. Second one is the middle layer, that is the cell wall. And third one is the innermost, and that is the cell membrane. We just mentioned that the cell wall is absent in only Mycoplasma, rest all have cell wall around the cell membrane. Though each layer performs a specific function, but they act together as a protective layer or a covering. Each layer performs a specific function, but they act together as a single protective lining or covering. Okay. Here we are going to add that also, that bacteria are classified into two types based on the difference in the cell envelope and the stain that it takes up. Okay, I'm going to mention that point here. We have already covered the stain part. Bacteria are classified into two types based on number one, difference in cell envelope. Number two, staining procedure. Staining procedure is just the same that we have covered. It's the staining procedure gram positive and gram negative. And difference in the cell envelope. What is the content made up of? What is the composition of the cellular envelope? Now, Next important point is prokaryotic cells have mesosomes. What are mesosomes? These are the extensions of plasma membrane into the cell. What for? These are basically responsible for division of the cell membrane and transport of material. They are in the form of, appears in the form of lamellae, vesicles and tubules. Clear? These are in prokaryotes. Then moving to the next one, the next point. They have chromatophores. Chromatophores. These are the internal membrane systems which contain pigments. Pigment associated areas. Okay, so these are the internal membrane systems. These are extensive in photosynthetic bacteria and cyanobacteria. These are extensive in photosynthetic bacteria and cyanobacteria. Right? Another feature that is quite prominent in prokaryotic bacteria are, that is this mobility. We say the bacteria or the prokaryotic cell, they show mobility. Why? Because these are provided with thin filamentous extensions which we called flagella. Okay, and each flagella, it has three parts. One is the filament, another one is the hook, and the third one is basal body. Another specific feature which is not flagella but are known as pili and fimbriae. These are surface extensions. Surface extensions and are brush-like. Like we talk about a toothbrush, they have a certain small bristles onto it. These give brush-like appearance. These are not concerned with the motility of the cell. These are not concerned with the motility of the cell. Rather, these are responsible for conjugation as well as in reproduction. Where two bacteria come closer of opposite strain and they show conjugation of the body, over there these pili and fimbriae are responsible. Clear? Then moving next to the other features which are found in the part of a prokaryotic cell. We first of all describe the cell envelope in detail. It is basically, as mentioned, made up of three components. right? So in these three components, the first one, which is called glycocalyx, it is basically a mucilaginous covering which is mainly made up of polypeptides, which are rich in amino acids, so it protects bacteria cell wall, because it does not allow it to dissolve in lysosome and other harmful chemicals from dissolving in other harmful chemicals. Okay, it has several other functions also: protection from phagocytes, protection from toxic chemicals, protection from viruses, protection from desiccation, protection from virus, protection from phagocytes that is being eaten up, right? This is what the glycocalyx. It is a sticky character, which is allowing bacteria to maintain its shape and is not lysed easily. Then is the cell wall. Cell wall, it is rigid solid covering. Rigid, solid, maintains shape. What else? Provides structural support. That's why we say, talk about cell wall in plants, no? It maintains shape, it is rigid, solid, provides structural support. This solid does. Where to the cell? It lies between the plasma membrane and the glycocalyx. Okay? And it's basically the one which is a channel for the transport of material in and out. Okay? Third one is membrane. The cell membrane or the plasma membrane. It's the similar membrane, the covering of the cytoplasm. Components will remain the same, made up of lipid and protein content. Right? Holds the receptor molecules for detection and responding to different chemicals of the surroundings. It holds the receptor molecules for detection and responding to various or different chemicals in the surroundings. Okay, so this is the cellular envelope in detail. Now we are moving to what else is there in the cytoplasm. Now moving to cytoplasm, which is known as crystallocolloidal structure. Cytoplasm of a prokaryotic cell. We say it is crystallocolloidal complex that forms the protoplasm excluding the nucleoid. Basically क्या बोलते हैं इसको? Protoplasm excluding nucleoid. Why? Because it is not a well-defined region. It is only a part of it. It is granular. Cytoplasm की nature क्या है? It is granular. Why? So many ribosomes are present in. Not much of the organelles are there, but yes, so many ribosomes are there. And membrane bound certain other like sap vacuole is present, gas vacuole is present, ribosomes, sap vacuole, gas vacuole, all these are present and make it granular in appearance. Moving to ribosomes. These ribosomes are found attached to the plasma membrane of the cell. These are first of all small, membraneless, submicroscopic, structures or units. Okay, these are found attached to the plasma membrane, okay, of the cell. These are of 70S type. What is the 70S type? See, S denotes the sedimentation coefficient. Okay, basically sedimentation coefficient or Swedberg's number. Each ribosome has two subunits, the bigger one and a smaller one. Okay, now when we are saying that this is a 70S type of a ribosome, it means its largest subunit is 50S and the smaller subunit is 30S. Together they make 70S, not 80S. No, 50S and 30S subunit does not make it an 80S, it makes it a 70S. Alright, and each ribosome, when it takes part in protein synthesis, it has to synthesize a chain. Okay, these are fixed ribosomes, they form a chain. 4 to 8 ribosomes are attached to a single strand of messenger RNA and they occur in a helical group. So we call it a polysome. When they attach together in this chain on an mRNA, this is mRNA strand. What is mRNA? Messenger RNA. These are in the form of a polysome. That means grouped together. Okay, here they are being used, or this process is meant for translation. What is translation? Translation means protein synthesis. This all we will do in class 12. Fine. So what we need to know is, here it is 70S. It has two subunits, 50S as well as the 30S subunit. They attach together by a strand of mRNA and they form polysomes. Okay, and they help in the translation process. That means they help in the translation of this mRNA into proteins where they are attached. Alright, clear? Now moving to inclusion bodies. Inclusion bodies क्या होता है? These are non-membrane bound structures okay, non-membrane bound structure which are freely lying in the cytoplasm. Okay, freely lying in the cytoplasm. Why? meant for the storage of reserve food material. Whatever is the reserve food material that is stored here. So they are basically categorized into phosphate granules, then glycogen granules, then cyanophycean granules, depending upon what they have to store. And another important thing, gas vacuoles are also found in cyanobacteria, which is known as a blue-green bacteria, green and purple photosynthetic bacteria too. All right. Now, this is about the ribosomes. Then we say chromatophores. Chromatophores are also the internal membrane systems which are present. They are typically non-unit, non-lipid and proteinaceous. They are in this form, especially when they are present in the green bacteria. Okay, and in purple bacteria, they are typical. That means these chromatophores are observed, these are seen. They have photosynthetic pigment, bacterial chlorophyll. They contain the photosynthetic pigment. Which is carotenoids and bacteriochlorophyll. Moving to the nucleoid region of this prokaryotic cell. Nucleoid material means the genetic material. Okay. This consists of a single circular strand of DNA which is supercoiled. How? Supercoiled with the help of RNA and polyamines. Polyamines and forms nearly oval or spherical complex. So basically, these polyamines or nucleoid proteins, they are different from histone ones, histone and non-histone complexes. Here we say polyamines, they are just concentrated foldings nearly 250 to 700 times, and they form solid structures. And this DNA of a prokaryote, it is considered naked because of its non-association with histone proteins. DNA is considered here naked because of no association with histone protein. Okay, now what is this naked and covered part is? When we say that this DNA, it is present in this form, then it is further coiled together, it forms this unit where there is small structural protein coat present where it is intertwined, and ultimately it forms a structure. So histone core is where the 8 amino acids are present, and over there DNA is wrapped. DNA is wrapped over the histone octamer. That is mainly present in a eukaryotic cell, and we call it, since it is a wrapped structure, this one, we call it, it's a completely covered structure. In case of nucleoid, it is naked because of no association with histone. Histone octamer is not present. So in which form this DNA is present? Naked. Alright, this is it. The next one is plasmid. It's also very important. Self-replicating extra chromosomal segments. Self-replicating extra chromosomal segments of double-stranded, DS stands for double-stranded, circular, naked DNA. Now, what is this feature? This is responsible for carrying certain characteristics of the desired gene into the alien DNA. Maybe I want to transfer this particular gene characteristic, this sequence, from this host to this recipient. So this is a host cell gene specific palindromic sequence, means a sequence of the bases. This plasmid will act as a vector or a carrier to transmit this desired set in its body to this recipient. This is how it makes certain antibiotic resistant genes also, drugs also. It is very, very important because of its this plasmid nature makes it a very special feature or a characteristic feature of all the E. coli or bacteria, because this is something which is acting as a boon, right? So it acts as a factory or a machine or a unit which act like a vector or a carrier in transmitting the desired set of genes from the host to the recipient. Alright, this is about plasmid. Then we have covered inclusion bodies, gas vacuole, food reserve, right, Synophycean starch, flagella, we have covered pili and fimbriae, we have covered. This is about prokaryotic cell.